Thank you for your interest in my poster entitled Rapid and Reproducible TAB and NAB AAV assays across serotypes species to support global preclinical programs. My name is Quentin Osseman. I'm principal scientist in immunogenicity method development at Altasciences, and I will give you a brief overview of the work we have conducted. Adeno-associated viruses, or AAV, are viral vector for gene therapies. The pre-existing immune status evaluation is required to support global preclinical programs. A total antibody, TAB, or a neutralizing antibody, NAB cell-based assay, were developed by Altasciences. The AAV gene therapies depends on vector transition efficacy, which can be reduced in presence of anti-AAV antibodies. For this purpose, fast and reliable TAB and NAB assays were developed and optimized, ensuring a transferability to different AAV serotypes and also animal species. The TAB assay used AAV capsids labeled with either biotin or ruthenium, leading to the formation of and detection of bridging complexes. The NAB cell-based assay measures the viral transduction of HEC293 T cells with bioluminescent expressions. In the presence of neutralizing antibodies, the viral entry will be inhibited, preventing the luciferase expression. The NAB cell-based assay was evaluated with different assay parameters to be standardized. First, results collected showed that compound C enhanced significantly the transduction efficiency of HEC293 T cells as early as 24 hours post-transductions. Second, the absence of preceding steps enhanced viral transduction and then luminescence expression. And then third, the combination of compound C stimulation and the no-sinding enhanced the transduction efficiency of HEC293 T cells using different AAV serotypes. The tables indicate assay parameters for each assays. They have different minimum required dilution, MRD, and sensitivity. Both assays require the same amount of sample volume and have the same precision of titer. The rest of parameters evaluated like specificity, hook effect, homogeneity, and other meta-acceptance criteria. The graph presented the screening status of AAV8, TAB, and NABs using 12 NHP serum. Results are in alignment with published prevalence of AAV8 in non-human primate serum lots for total antibodies, TAB, and NAB. Similar data have been collected with the AAV9 serotype. In conclusion, two bioidentical assays were successfully developed and standardized. Both assays provide data within 30 hours and showed also transferability with minor adjustment to other AAV serotypes, over animal species, and different Altasciences biolab sites. The AAV9 NAB methods was used for different study sample analysis, allowing the screening of more than 200 animals. Overall, the TAB and NAB assays offer complementary insights. The TAB assays provide a broad overview of binding antibodies response, and the NAB assay measure specifically the functional neutralization capacity. Together, they enable a comprehensive evaluation of immunity.